All right, welcome everyone to our very first Advanced Topics Bioinformatics Bootcamp. We're going to be talking in this workshop about single-cell RNA-Seq analysis. And my goal is to make it so that you feel like you've got the knowledge and the skills to apply this technique effectively in your own research. All right. Did someone annotate on the... Okay, I'm gonna go ahead and disable annotation for others so that we don't have any more of that. This part's gonna get cut out of the recording. All right, so now we should be ready to go. So without further ado, let's go ahead and jump in. So why are we doing this workshop? Well, I'm going to tell you what I tell people when I do a workshop typically, and there's going to be a bit of a twist for this time around. But essentially, those who know bioinformatics will be more effective at research, more competitive for jobs and for funding, and more flexible and adaptable to changes in their field. But you could say this really about any sort of bioinformatics or really any sort of data science that you learn. Why this workshop in particular? In this workshop, we have a more important goal than all of that put together, which is to learn single-cell RNA sequencing. And I think as we go along, you'll kind of understand why I emphasize this so much. I mean, I should say why I emphasize it so much. I mean, look at this. This is beautiful. There's really no way to overstate the transformative power of this technology on our entire understanding of the biological world. I mean, I really have no way to overstate that. And the point of this workshop isn't just to prepare you for your career or help you do good research. It's specifically because I believe every single person who is exposed to this technology, who has the ability to analyze this data, is going to basically open up new biological insights that we never could have found otherwise. And I think by the end, you'll probably agree with me. Now, the truth is that from this workshop, our goal is a bit simpler. We're really just trying to get you to the basic steps. The first thing you're going to learn is how to perform basic data preparation for single-cell RNA-Seq. We're going to move on to manifold learning techniques for analyzing cellular relationships in this kind of data. We're going to be able to find and interpret biologically meaningful patterns in these data sets. We're going to integrate single-cell data from different batches, studies, and modalities. This is actually a really important technique, and this is part of why I want you to have version 4 of scRNA, because they've got some really exciting tools that help you do this more effectively. You're also going to learn how to present the results of a single-cell RNA-Seq analysis with publication quality figures. By the end, you'll have the skills and the confidence to use this technique in your own research. So while we're not going to get you to the point where you could do this, and this is very impressive and all, we're at least going to get you to the point where you can do this. These are the basic things that you're going to learn how to create. You're going to learn basic quality control, basic manifold learning, how to do basic plots and visualizations, and how to create cluster analyses. This is what, in essence, you need to do a basic level single-cell RNA-Seq analysis that could be published. Okay, so what's the structure of the workshop? Most of the sessions are going to be about an hour of lecture and 30 minutes of hands-on activity. For various reasons, this one is going to be mostly just lecture. I have a lot to get through, and at the end, we'll do a short activity. But most of them are going to be a bit more even. The lectures will be recorded. I've said that a few times, but I want to just keep iterating that because I want you to know that you need to keep your camera off and your microphone off if you do not want to be recorded. This is the basic outline of what we're going to talk about. So today's the orientation. We're going to go through those other modules I had mentioned previously. And then the last module is actually a final project where we're going to be finding metastatic subpopulations in actual patient tumor data. Now, you don't have to do this final project. And in fact, if you don't have the time, I would encourage you to skip it. What we're doing here is we're offering you the opportunity to do a very real world single-cell RNA-Seq analysis, present it to us, and then get real-world feedback. Because I'll tell you what I'm going to do. You give me your analysis notebook or your analysis slides, and then I'm going to pretend like I'm your PI, right? So like, let's assume that a lot of you are probably students or postdocs. I'm going to pretend like I'm your PI, and I'm going to respond to this the way that my PI responds to this, and the way that other PIs who themselves don't have coding ability respond to bioinformatics, which is, does this show me the biology? And what I'm going to do is I'm going to give you that feedback, and then I'm going to help you understand from a bioinformatics perspective how to tweak your analysis if it needs to be tweaked in order to meet the needs of someone who you might want to present it to who doesn't have that coding background. Same thing holds true for paper publications as well. Most of your reviewers will probably not have coding experience, depending on where you're going. So it's optional, but I've been to a lot of workshops. I've never had someone offer me the opportunity to get that kind of feedback. So it was something I wanted to offer you. And if you want to take advantage of it, you are more than welcome to do so. So how do you succeed in this workshop? Well, the first step and the most important is respect the prerequisites. So I sent a few emails about this, and I've said this a couple times, but you really expected at this point to have basic familiarity with R programming and RNA-Seq analysis. If you're here, I would really like it if all of you have at least the R programming, but preferably both experiences. And if you don't have it and you want to do the workshop anyways, that's fine. I mean, I'm not like the police. You could catch up in the next week as well. We did a workshop last semester where the link to it is actually here. And you can go and catch up on anything you've missed. One thing that I don't think I had mentioned previously, which I'm just going to say, is if you go to the GitHub page that was sent in the email previously, you'll be able to find the PowerPoint that I'm showing you right now. So if you go there, and I'm actually going to give you the link. So let's go ahead and do that now. I'm going to put this in the chat just so that all of you have it right at your disposal. So let's go ahead and do this chat. All right. So I've just put the link in the chat. Make sure you're checking that out and that you're following along in the PowerPoint if you'd like. Later on, you're actually going to clone this repository on your own computer. So hopefully you've all got Git, or even if you don't, it's okay. You can actually download it here as a zip file. So there's two ways you can get this, or you could simply, I think, download this file individually if you want. So you can download it individually if you want. This is the module 1 PowerPoint. Okay. So now that I said that.
Hello, Henry. Hello.
Hi, go ahead.
Hello. Yeah, I have one question here. Like the present version of R that I have installed in my system is 4.0.3. Okay. So it is not supporting Bioinformatics, which actually convert the IDs. So could you send me a solution? Could you tell me solution of it?
So I'm not sure. I'd have to take a closer look. You're more than welcome to come to the office hours, and I'm happy to help you there. Or perhaps Aola or some other of the students who've helped me with this could possibly help you in the chat. But I would say for right now, probably best to leave that for outside of the lecture, just for the sake of time.
Okay.
All right. And in fact, for future questions, I'm going to have a question section at the end. But for future questions that come up during the presentation, it would be great if you could put that in the chat. And actually, the other students who are here helping me out will probably be able to help you with that question. So please put those in the chat, and if they don't get to them, I'll make sure to get to them after the presentation. All right. We were going to offer Data Camp Classroom, but we had an issue setting it up. To be frank with you, you don't need it for this. There's really not a whole lot of relevance of Data Camp Classroom for what we're doing here. It's really more for the basic R workshops that we give. It's just nice to offer when we can. If you find yourself needing some help with some of the more basic tools and techniques, though, Data Camp is a great resource. And probably by next week, we'll have access to Data Camp Classroom for everyone. But again, it's really not necessary for this particular workshop. You can also look at outside resources like Codecademy, RStudio Books, and in particular, with learning single-cell RNA-Seq, there is a large number of tutorials and YouTube videos out there where you can go and you can see someone else explaining the things that we're going to be talking about, and then getting it from both perspectives so that you can get a more rounded view on how to do your analysis. So I really encourage people to do that. Don't consider us to be your only resource for learning, because we don't have all the answers. All right. Finally, communicate with your instructors. I've got my office hours, which you can attend over Zoom. So I use Calendly to set these up. It basically means that you can sign up for an appointment. And once you sign up for an appointment, no one can take that from you. We have an exclusive, one-on-one 30-minute block where I can help you with any issues that you're running into. So if you find yourself running into issues or having questions, feel free to, at any point, come and attend those office hours. Okay, so Shibo has let me know that the link for the fall 2020 R programming and RNA-Seq analysis is apparently not working. So let me go ahead and share the up-to-date. Actually, Aola, if you wouldn't mind sharing that, that would be great. The link for the R programming and RNA-Seq analysis workshop. I'll make sure to share it once we get to a break in a little bit here.
Okay, sure. I got it.
Okay, thank you so much.
No problem.
And I know I said I wasn't going to stop for questions, but I want to address this one really quickly. So Nazia asks, are those for students only at your institute? And it's not. It's open to everyone. We don't have any requirement that you be from our university. So this is open to everyone who wants to attend, and those resources are open to everyone. We want to really encourage kind of a worldwide sort of bioinformatics literacy. I mean, that's kind of one of the main goals we have. All right. So about our club. So this is brought to you by BIG Club, that stands for Bioinformatics Interest Group. We're a club at UT Health San Antonio promoting student engagement with bioinformatics. Now, to join this club, though, you do have to be a student at our university because it is a university club. You don't have to be a student to be in our workshops, but to join the club, you'd have to be a student. Our next meeting is going to be February 23rd from 6:30 to 7:30 p.m. And if you are interested in coming to that, just shoot me an email and I'll make sure you get added to the list. Okay, let's go ahead and jump in. So to understand the purpose and kind of the majesty, in my opinion, of single-cell RNA sequencing, it's important to first understand its precursor, bulk RNA sequencing. So let's start with this kind of simple idea here. mRNA holds the answers. The answers to what? Well, biological states. And when I say biological states, what I'm really talking about is everything from tissue types to drug treatments to survival and to genotype. These are the phenotypes and the behaviors and the conditions that we try to understand and unravel as biologists. Now, one of the things we've come to understand is that these biological states have a correlation with the production of RNA. So perhaps gene expression, splicing. We know that these states are represented at the level of the RNA. So what do? We measure the RNA. We use RNA sequencing to do this. And ultimately, the goal of this is to learn about the biological states from which that RNA arose by measuring things like gene expression, splicing, gene fusions, and transposon expression. Those are what I would say are the main things we tend to look at in RNA-Seq data. So a little bit about how this works. Typically, you're going to start with some mRNA. You're going to convert it to cDNA, fragment it, and then proceed to sequencing. So when you sequence a fragment of cDNA, or when you sequence a sample of cDNA fragments, what does that look like? Well, I'm going to actually show you the file this gives you back. This is a FASTQ file. More accurately, this is the first like 15 lines or 20 lines of a FASTQ file. And this can be a bit daunting at first. This is an actual FASTQ file from one of the experiments I was working on a few weeks ago. But I'm going to help you understand what this is telling you. So first of all, you have to understand that FASTQ files have a repeating quartet structure. So each of these groups of 4 lines is a read. This is read 1, read 2, and read 3. And this is, again, out of like millions of reads from your RNA sample. So let's take a look at one of these reads and break this down. The first line in the quartet is the read ID and the sequencing info. So this is telling you information about which read this is, its unique ID in the experiment. It's also telling you information about the sequencing machine and where in the sequencing machine this read was located, which is this sort of, I'm going to switch to laser pointer. It's this sort of address up here. You don't have to know how to read that. And in fact, you'll probably never directly do anything with FASTQ files by hand. It's just interesting to know what they mean. The second line is the actual sequence of the cDNA that was sequenced. So your G's, C's, A's, and T's. The third line is a placeholder. We actually don't use it for anything as far as I know. And a lot of people have been talking about ways to just get rid of it. The fourth line is the quality score of each base. So for every base here, so for example, this G, there is a corresponding quality score. And it's encoded with these symbols and letters. So this would be the quality score corresponding to this G. This one is the one for this G, A, and so on and so forth. And you don't have to, again, you don't have to do this by hand. The programs you use will automatically use that information. But the point of this is that whenever the sequencing machine says, I think this is a G, it also says, this is how confident I am that this is actually a G. And that's what the quality score is essentially telling you. So again, you're not going to use that by hand, but it's just interesting to understand how the data looks when it's in a raw format like this. All right, so the next step after you get these reads is to align them to the genome. Now, when we're talking about RNA-Seq, typically you're going to find that your reads are aligning to exonic regions of the genome. So that's why we see these clustered like this. That's implying that these are 2 exons here and here in some gene. So let's take a closer look at that. A mentor of mine actually made this animation, so I want to make sure he gets credit for it. But I use it because I think it's one of the best that I've ever seen for explaining how this looks. So this is an idealized model of a gene. It's got promoter, it's got exons, introns, and intergenic regions at the end. It's also got a 5' and 3' direction and a transcription start site. When we align our RNA-Seq reads to this, typically we're going to focus on the reads which align to the exons. This is what that would look like. So you can see here the reads aligning to some location on the exons. Now in the mature RNA, typically the introns are going to be spliced out. Typically you're not going to have any promoter regions or intergenic regions. And so we would expect to see either no reads or only a few reads aligning to those spots. But of course, there's situations where that's not always true. And RNA-Seq can be an interesting way to assess that as well. All right, so here's a little bit of a summary. In general, what you need to know is that we're typically concerned with what's going on at the exons. And if you want to assess non-coding RNAs and small RNAs like microRNAs and PWE RNAs and that sort of thing, you'll typically use different kinds of RNA sequencing. Another issue you have to deal with in RNA sequencing is the fact that in a lot of cases, the mature RNA has the introns spliced out, which means your exons are stitched together in that mRNA. And that means that when you do your sequencing, you'll have a lot of reads where half the read aligns to one exon and half the read aligns to another. And this is why you need to have a splicing-aware aligner like STAR that is able to deal with this and annotate it. So I'm actually going to show you now a demo of a program I use all the time called IGV. If you've never used it, I would highly recommend downloading it. It's free. It's open source. And it's probably the most useful tool that I've ever found for looking at sequencing data and assessing its alignment. So, okay, so what we're doing here is we're loading up the genome. And you can see here now, this is what it looks like when you've got the genome loaded up, specifically the human genome, HG38. In this particular case, HG38 is one particular version of the human genome. It's the newest version. We can also choose HG19, which is an older version of the genome. And there's also other species. If you come down here, you can find all the species and all the versions of all the genomes that are available. But I typically prefer HG38 just because I think it's the newest and it's the one that I'm just most used to at this point. All right, so let's say you want to look at a particular region. You could come down here to a chromosome, and when you click on this, you can see the ideogram for the chromosome across the top. And this is telling you where the short arm is, where the long arm is, and the different bands that exist in that chromosome. If you want to zoom in on a particular region, it's pretty easy to do. Let's say I want to come to this band here. I can just drag this area. Oops, there we go. And I can see what's going on in this region. If I want to zoom in further, let's say I want to zoom in on a particular gene. None of these are particularly exciting genes. I'm going to go along here until I find one. Ah, there we go. Jared2. Let's go ahead to Jared2. So I just zoom in here, and I can keep zooming until I've got it displayed the way I want. Now, one of the biggest and most important features of IGV is that you can easily look at these splicing isoforms for each gene. And so I can see here the different isoforms that are available for Jared2. And I can also see very easily where the transcription start site is. So I know it's on the left side because the arrow points to the right, which means it starts on the left. And if I zoom in even further, I can now see the exons and the sequence is starting to appear. So let's go in one more time. And now I can even see the amino acids these sequences code for. So this is a very powerful tool. And the main thing you're going to do with it is you're going to see how this relates to some alignment file that you have generated. So this actually comes from an RNA-Seq experiment that I'm currently working on. We usually use BAM files. That's just an alignment file that is very compressed and memory efficient. So I'm going to load this up. And now I can see here, these bars are the reads. And so these are the reads that aligned at this exon or aligned to this location. I'll zoom out a little bit because it's kind of hard to see. Okay, so what is this telling me? Well, on the top, I can see for every nucleotide, the number of reads that aligned at that point. So basically not a lot of reads aligned here, and most of the reads aligned right around here. And then once you hit the intron, not a lot of reads again. So again, with RNA-Seq, you're typically seeing things in the exons. So let's go to a place where we can do a nice example of the exon junction issue. We'll come over here, since this is going to be a place where exons are very close together. So let's take, for example, these two exons. I can see that I have a lot of reads aligning in these two spots, and I have a lot of reads that are split. So for example, this read, half of it aligned here, and then the other half, if you trace this all the way, aligned here. Actually, you can't see that one super easily. Let's find a better example. This one and this one. So you can see where the split in the read occurs, and star, which is the aligner I used, annotated where that junction is occurring. If you look here at the junctions track, you can see those junctions as well, being represented as arcs. The other thing you can do, which is pretty cool, is you can come down here and make a sashimi plot. Now, a sashimi plot is a way of representing splicing junctions in a sort of summarized pictorial format. So we can see here there were 70 reads that had this particular junction. And that's what this is essentially telling us. It's also an interesting way to assess alternative splicing. So for the sake of time, I'm not going to show this, but if you go to BRCA1, there's a lot of alternative splicing. If you make a sashimi plot, you can basically see when an exon is being skipped or when it's being included. All right, so that's IGV. If you've never used it before, I highly recommend checking it out and giving it a try.
Henry, Wes had a quick question. If you are using custom genome, let's say we have added a fake chromosome to our GTF and FASTA, can IGV load our custom reference genome to see how reads align to our inserted gene in our transformed cells?
Absolutely. And that's actually one of the best features of IGV. I use it for that, for looking at custom genomes all the time, actually. So what you can do is you can come here to genomes. You can load a genome from a file, or you can create a genome file. And this is essentially the way that you can make custom genomes. And essentially, IGV is going to index them so that they can be displayed like this. Because there is, in fact, a common notation that all genomes have. All you would have to basically say is where the chromosomes are. And so, for example, if you've got a viral, if you've got some sort of, I don't know, construct, you could basically say construct A as the name of your chromosome, and then just give the sequence. Okay. So great tool, use it all the time. Really fantastic for assessing the quality of your alignments and for really seeing any sort of data where it aligns to the genome. Okay, so what is the next step? Well, the next step is to quantify. What you need to do now is basically figure out for every gene, how many reads aligned to that gene. This is the result of quantification. We've got what we call a gene count matrix. Genes are the rows, and so there's about 35,000 or more of them. The columns are the samples. So let's say in this data set, we had 9 samples. And so what we're learning is for every sample in every gene, we know how many reads from that sample aligned to the gene. And that is our read count. That's why it's called the read count matrix. So I know for sample 1589T in gene ENSG0000227.232.5, there were 85 reads which aligned. So let's say now we've got a bigger goal. Let's say we want to ask the question, how do Alzheimer's patients differ from healthy controls with respect to gene expression? And in fact, phrased another way, we're asking, what are the differentially expressed genes between these conditions? This allows us to say things like, P53 is overexpressed in Alzheimer's disease brains. So it's that kind of analysis where we can come up with that sort of data. We use DE-Seq, EdgeR, sometimes Lima if you want, to do that sort of analysis. I typically do DE-Seq too. And you'll generate plots such as volcano plots and MA plots. GSCA is another technique we'll often use here. And you'll also make heat maps. So I'm actually going to show you a heat map from an analysis I did in Alzheimer's patients and healthy controls. So basically, these are Alzheimer's patients, and this is compared to healthy controls who had no cognitive impairment. That's NCI, no cognitive impairment. And so as I start to drill down into the data, I might start looking at individual genes, and I might say, well, I noticed GSTM5. GSTM5 is interesting because it's involved in oxidative stress response. So I might hypothesize from this that in Alzheimer's disease, you have an increase in oxidative stress response pathways. And then I could do a pathway analysis to help me demonstrate that. So looked at another way, this is sort of a box plot representation of that gene's expression between conditions. You can really see it's quite highly overexpressed. So in summary, with bulk RNA-Seq, which is what you've seen so far, you can easily ask a lot of questions. You can ask, what are the pathways that are overexpressed? What are the biological processes and drug targets that are changing between conditions? What gene expression patterns associate with survival? And which ones associate with genotypes? So if you had the clinical data, you could easily ask those last two questions. But there's questions we can't ask. And this is essentially why you're here and why single cell exists. Because there's a lot of stuff we can't know with this kind of data. We can't know, how do cellular populations change between these two treatment groups or conditions? How do cell types, specific cell types, show differential expression? So we're kind of asking the question here, if we see a change in the expression of a gene in bulk RNA-Seq, we have no idea whether that gene is actually overexpressed or whether it just happens to be that the cell type that expresses that gene is more highly represented in the sample. There is no way to tell the difference with bulk RNA-Seq. We can also ask questions like, are there cell subpopulations that are capable of surviving? So in the example of Alzheimer's, this would be neurons that don't die when the other ones do. And are there more vulnerable subpopulations? So again, these are the kind of questions we can't ask because we don't have access to that kind of level of resolution. So basically, the way I see this and the way I think most people see it is that there's a range of suitability for bulk RNA-Seq in different experiments. Clonal cell populations, sorted patient cells, those tend to be fine. But when you get to the realm of organoids and even worse, postmortem tissues and tumor biopsies, at that point, there is a hard limit to how much you can learn from bulk RNA-Seq because you don't know what's going on at the level of cellular subpopulations. Introducing single cell. Every cell is a unique sample. So what does this mean? This is bulk RNA-Seq. We've already talked about this. Essentially, you're taking a heterogeneous tissue like a postmortem brain, you're mashing it all up, you're extracting the RNA, and you're getting one gene expression profile for that entire sample. And you have no idea how cellular subpopulations are contributing differently to that expression profile. All you see is the one expression profile. But with single cell, every individual cell has its own distinct expression profile. And the reason this works is because every read that you sequence, you have the barcode that tells you what cell it originated from. And so you can tell which reads came from which cells, and therefore you can see for each cell, what genes is it expressing and how highly. There are also limits to single cell, which we'll discuss next week when we get into quality control. But for right now, just know that this is in essence what it's doing. And hopefully you can start to see why it's so amazing. So to help illustrate this, I'm going to show you a paper that really inspired me. And essentially what this paper does is it does a single cell RNA-Seq analysis. It's almost entirely bioinformatics, and yet it still manages to reveal an incredible amount of novel insight into Alzheimer's disease and also get into nature. So let's jump in. I'm just going to kind of quickly go through this and hopefully illustrate for you some of the things that I'm not necessarily going to teach you how to do, but the skills you gain in this workshop will basically enable you to do in the future. All right. So this was their design. You had half Alzheimer's, half healthy controls, half male, half female. They took the prefrontal cortex. It was post-mortem frozen tissue. They isolated the nuclei. So this is actually, technically, this is single nuclei RNA sequencing, not single cell. But for the purposes of the paper and for what is in the scope of this workshop, just kind of consider them to be the same with some minor technical caveats. If you ever find yourself doing single nuclei RNA sequencing, I would say that's the point where you should look a little bit deeper into what the differences are. All right, so they took these, they found about 80,660 cells, and this is the 2D embedding of those cell gene expression profiles, which this is something you'll learn how to do in our workshop. The next thing that they did was they clustered the cells, and then they did cell type annotation. And essentially what this means is that they're using an unbiased approach to finding groups and groupings of cells. That's the clustering. You'll learn how to do that with us. And then they did cell type annotation. So basically saying, I've got cluster number 87. It expresses, GFAP, and it expresses some other microglia markers. Therefore, I think cluster 87 is a microglia cluster. And there's various tools and approaches you can use to do that. This is something we're going to touch on as well. Now, this is their figure 1. It's really beautiful. It's kind of, when you see this, it's kind of no wonder they got into nature. It's just such a beautiful way to represent the data and provide the biological meaning behind something as complex as a UMAP or t-SNE. They're also showing you here the markers that they were using to find the different cell populations and cell types in their data. Okay, so I'll say something. I'll say that when I see papers that do single cell, a lot of them actually stop here. They don't really go any further because a lot of people have the idea that this is, in essence, the end goal of single cell is to make a plot that looks like this. But that's really not true. This should be more like figure 1. This shouldn't be your final figure. This is where you start, not where you end. And so what was really special about this paper is they said, we're going to make this, and then we're going to show you all the amazing things you can do after you've gotten to this point. And they developed a lot of new methods and a lot of new approaches, which I'd like you to see and to hopefully, like me, be inspired by. All right. So the first thing they did was they took the clinical covariates that had continuous values. So these are from IHC experiments, as well as assessment of patient cognitive performance. Basically, for every patient in their data set, they have a score for each of these factors. And they were able to very easily see the Alzheimer's patients were different from the no pathology patients with these factors as the differentiator. And in fact, you can see, for example, global cognitive performance level is its lowest in these Alzheimer's patients and highest in the no pathology patients. So this was a really exciting first step for them because this is, in essence, a trick you can use. And it's one that I've only recently begun to appreciate in the analyses that I do. But essentially what they've done here is a version of an unsupervised machine learning analysis. They started out, they only had 2 labels. They had no pathology and AD pathology. But by making a heat map of all of these continuous values between all of their patients, they were able to implement hierarchical clustering. And that's what you're seeing here. That's what this dendrogram is showing you. It's showing you hierarchical clustering. And they were able to identify a novel grouping of their AD pathology patients into early stage and late stage. So this is really exciting because they are basically showing here that you've got some patients who have Alzheimer's who really are quite different from the other patients who have Alzheimer's. And this is actually going to translate into the rest of the analysis that they do. So First goal was to find differentially expressed genes by Alzheimer's disease stage. This is the early versus no pathology comparison. Now, what you're seeing here are the genes that are overexpressed in the early pathology versus no pathology group. And you can see here you've got excitatory neurons, inhibitory neurons. So you can see for each of these cell types what those are. And this is starting to evoke a really interesting idea that they're going to chase for the rest of the paper, and ultimately is going to be the crux of their paper, which is that you have some genes that are overexpressed in multiple cell types. And this evokes this idea of a pan-tissue marker of Alzheimer's pathology. But this goes into overdrive when they now look at the late pathology versus early pathology comparison. This is that heat map now. So basically what you can see here is that there are a lot of these sort of pan-tissue markers. So HSP90AA, all 6 tissue types show overexpression of this gene in late versus early pathology. This is a really exciting idea. And there's one of the things that was the biggest selling point for this paper is that they were identifying these pan-tissue markers of the Alzheimer's late pathology. So they did the same with the underexpressed genes, and wasn't quite as exciting, but still interesting. The next thing they did that was quite novel is they looked at the number of perturbed cell types as a predictor of the fraction of upregulated differentially expressed genes with respect to these different comparisons. So what does this mean? This is like, I found this figure very counterintuitive at first. I'm just going to break it down by looking at only one of these columns. So let's look at number 2 here. So we're basically saying here that in the late pathology versus early pathology comparison, about 25% of the differentially expressed genes were overexpressed in at least 2 distinct cell types. So that's why this is quite interesting, because this tends to suggest that the late versus early comparison is actually much more pan-tissue than the other comparisons they could have made. Okay, and this becomes more stark as you increase the number of perturbed cell types. At the end here, 5 and 6, you know, you're only finding the late versus early in these groups. So basically only in the late versus early comparison are you seeing these truly pan-tissue responses. And so then they wanted to say, well, all right, we're seeing this. What does this mean? So they did a pathway enrichment, and they found that this was largely related to protein folding, autophagy, apoptosis, heat shock, and the sort of stress responses that are often so interesting to examine in Alzheimer's disease. And this was actually, I'm in the biology of aging PhD program at my university. I presented this in our Biology of Aging Journal Club, where a bunch of people study Alzheimer's. And this was the thing that I think they found the most interesting, because these are new evolving theories about how Alzheimer's happens. And this was the first time I think that really anyone had shown in this sort of unbiased, you know, high throughput sequencing based approach that you were having these processes occurring this way. So it was very exciting. And I think this is one of the major selling points of their paper. But it's also a novel analysis. I have not seen anyone do this before or since. The next step that they took was to do something called subclustering, which is also something we're going to teach you how to do. Essentially, when you take one of your bigger clusters, rerun it through the pipeline, and then see what subgroups exist within it. In this case, they were looking at excitatory neurons, and they wanted to see were there any novel subtypes of excitatory neurons that they could find. And also, did any of them associate with pathology? So I'm going to show you the very first part of this figure. Since if you can understand this, you can really understand the rest of it. These are 3 of the most biologically relevant subclusters that they ended up finding in the excitatory neurons. And I know you don't see the labels here, but basically what this means is low amyloid, high amyloid, low Braak stage, high Braak stage, you know, decent cognitive status, poor cognitive status. So as you go from left to right on these 3, it just gets worse. what was really interesting is that you had these clusters which show enrichment of these sort of markers of pathology here. So cluster 4 has the highest levels of amyloid, the highest Braak stage, the lowest cognitive status. So this is, by all objective measures, this seems to be the cluster most associated with the Alzheimer's pathology. Same thing with cluster 6, except reversed. What was really exciting, and which they ended up exploring in another figure I'm not going to show you, is that, so this is male and female, that there's actually also an association in these two clusters with sex. And so that's actually playing into one of the theories that is very much emerging for how Alzheimer's works and why you see sex-specific pathology. And this was a very exciting way to demonstrate that. So we see this with the excitatory neurons. We see this with the other cell types as well. They also overlaid on top of this the other clinical covariates, such as the cognitive level and the amyloid, and these are sort of the continuous rather than the discrete. So these are all discrete and ordinal, and these are all continuous value covariates. And so we can see that this paints a really interesting picture of the subclusters. The next step for them was to actually take one pair of clusters and analyze them further using something called cluster marker analysis, which I'm also going to show you how to do. So you've got your two clusters here. One of them is enriched for Alzheimer's disease patient cells. The other one has low incidence of the Alzheimer's disease patient cells. So one of them seems to be more associated with pathology, whereas the other is less associated with pathology. If we look at what are the genes that these clusters overexpress compared to everything else, we can see what their, quote, cluster gene markers are. We represent that with a word cloud, where the size of the word represents the significance of overexpression. So basically what this is telling us is that this subcluster tends to overexpress SLC5A11, and this cluster tends to overexpress CAD-M2, QDPR, and the rest of these. They also want to ask the question, well, what are the pathways that these relate to? And they did that very effectively here. All right, last thing they did, or at least last thing I'm going to talk about, is they went back to the lab and they said, well, all right, we found these clusters bioinformatically. Are they really there, or is this just something that we can only show in, you know, kind of the nebulous world of computer stuff? So they took two markers, QDPR and CRY-AB. And they said, all right, let's do an IHC and let's see if we can find these overexpressed in the Alzheimer's compared to the controls. So that's what they did. So this is CRY-AB. No pathology is the first row. AD pathology, Alzheimer's disease pathology, is the second row. And we can see here that, in fact, CRY-AB does appear to be overexpressed in the disease compared to the control group. QDPR was even more stark. You can see that it's very strongly expressed in the AD pathology oligodendrocytes, but not very highly expressed with no pathology. So, in summary, this is an excellent paper, and it illustrates some of the most incredible and advanced techniques you can use to analyze single-cell RNA sequencing data. It also, to be frank, kind of tells you how you can get into nature with basically just bioinformatics. I'm not sure if this would still work in the year 2021. It might, but at the very least, I think there's a lot of excitement still about even just the bioinformatics around single cell. So, well, I'm not going to teach you that. What I am going to teach you is a lot of the underlying techniques that go into those sorts of analyses. So, I mean, for example, we're going to basically talk about quality control, dimensionality reduction, cluster marker analysis, feature plots, and other visualizations that you'll use, and essentially how to think about the analysis and interpretation of your data sets. So what is that going to look like? Well, let's now talk about the single cell workflow that you're going to learn. Don't worry about understanding all of this right now. I know it probably looks a bit intimidating, but you'll actually find that most of these steps can be done in like 1 or 2 lines of R code with pre-built functions that don't need a whole lot of modification. But I will walk you through how to do them, and more importantly, how to think about the results and how to interpret the results. The other nice thing about this workflow is that you can do the whole thing with R and scRNA-Seq. So that's the package that I typically use most when I do single-cell RNA-Seq analysis. I don't typically find any reason to venture outside of it unless I'm doing a more specialized or custom analysis. And in that case, it's usually pretty easy to do the analysis in another tool and then reincorporate the data back into my scRNA-Seq object so that I keep everything nice and together. So you can get scRNA-Seq by going to their website, which I'm just going to show you here. So if you go to Google and you just search scRNA-Seq, it's going to be the first thing that comes up. So they just updated to version 4 like last week or a couple of weeks ago. So it's really exciting. And that's part of why I want to teach you version 4, because they added a lot of new stuff and a lot of new tools that I think you should be aware of. And then you'll have the most cutting edge knowledge that's out there. All right, so also, I don't know if scRNA-Seq is actually the right way to pronounce it. I've just been saying that for like a while now. It's based on this artist. So however you pronounce the artist's name would be how you pronounce this. But I like saying scRNA-Seq better, so. All right, so you can install it by going here and basically copying and pasting. Pretty simple. You can even click this button, so you don't even have to do the Control-C, Control-V. And then there are lots of vignettes to learn how to use all of the different functions and approaches that are available in scRNA-Seq. The one that we're going to probably start with is going to be the PBMC 3K guided tutorial, because to be honest with you, that is going to give you a lot of the most fundamental skills and a lot of the intuition for how to think about the data. So I'm going to go through this with you, or at least a lot of the steps of it with you, and then we're going to move on to more advanced work later. All right. So let's see here. I'm gonna mute you. Okay. So first step, load the data. So if you go to GitHub, you'll find that you've been provided with the count matrices from Cell Ranger for one PBMC experiment and a script for loading the data. So get used to going to GitHub and then going to RStudio, because for the remainder of the workshop, we're going to do most of our work in the actual code itself. I want you to actually see how this is done instead of just hearing me go on and on about it. So if you haven't already, make sure you go to GitHub, go to the single cell RNA-Seq analysis workshop link. So I'm going to put this one more time in the chat. So I just put it there in the chat. And then go ahead here, and if you've got Git on your computer, you can go ahead and I'll do this afresh so that you can see me doing it along with you. I'm going to remove my previous version of this. So if you're on Windows, it would be the command prompt. If you're on Mac, it would be the terminal. You can go ahead and type git clone, and then copy this link right here, git clone, and then paste. So this is the first way, and it's the way that you should learn to do, because if you want to do bioinformatics and if you want to do any sort of computational work, eventually you'll honestly just have to get comfortable with Git. But if you don't have Git on your computer, or if this is not working for one reason or another, you can simply go over here to where it says code, click that, and then do download zip. And then it's going to download it all as a zip file, which you can simply open. All right, so if you did it the Git way, then it downloaded it to the working directory that you ran Git from. In my case, this was users/mille. So I'm going to come over here to my PC, going here to users, MILLE, and there it is. So this is what you should see here. Okay. Same thing, if you downloaded it from the zip file, you can just click on this and extract it. You can extract it anywhere you want, but I've extracted it now to my downloads folder, and it should look like this. So 2 ways to get here. Pick whichever one you prefer, but hopefully you can do the Git one. Okay. So now that I'm looking at this, the next step is going to be to open this up in RStudio. And the way I want you to do that is to go to the module, and for every module, there's going to be an R project file. This is what I want you to open, because this will allow you to directly have everything open in RStudio with all the correct relative paths and everything ready to go. So go ahead and double click on that. And it should, if you have RStudio installed, it should open it. So now that's what's happening here. So it's opened it up, and I can see I'm already in the correct working directory with all the files and all the data. If I click on this module _1.r, it'll open up the file that I've given for you to work on today. Okay, so let's say that didn't work. So I'm going to close this. I'm going to pretend like I wasn't able to do that. Like when I double clicked on that R project file, nothing happened. Okay. So now I'm just staring at a blank RStudio. This is what it looks like when you first open it up and there's no project selected. What I'm going to do is go over here, open project, and then I'm going to find that R project file, and I'm going to select it. So, you know, I'm not going to like come over there and force you to do things like this, but to be honest with you, this is, in my opinion, by far the easiest and most straightforward way to manage your work in R. And it's the one that is going to minimize the number of issues you run into as you attempt to run the code that I give you and which you actually create yourself. All right, so we've gone through a lot. At this point, basically, we're going to go through the script. But before we do that, I want to take 5 minutes, and I want to just quickly answer any questions which have come up. So if you've got any questions, feel free to unmute and ask them now. Or put them in the chat. That also works. And I'll also give a few minutes for people who haven't gotten to this part yet to catch up. While we're waiting, I'm going to also make sure I put the correct box link for the workshop from last time in. Okay, so I'm going to put this in the chat here. So this is the link to the box folder that contains all the materials for last semester's workshop. If you need to catch up on R or RNA-Seq, or if you felt lost with any of the stuff we did today, I would recommend going through those. We've got videos and exercises, and if you're not comfortable with R, I think it's a good idea to probably brush up on that. Again, I'm not going to make you do anything, but if you feel lost all the time as we're going through this stuff, it's not going to be good for you, and it's not going to be an enjoyable experience. Okay. And I'm also going to update really quickly the link in the actual PowerPoint itself to make sure it's the correct one. So in the future, that one should work just fine. All right, so with the last 15 minutes, let's go ahead through this here. Oh, okay. So Mauricio asked, are we going to see the analysis starting from FASTQ files? Next week, I'm going to briefly show you how Cell Ranger works, because essentially what you do is you just give the FASTQ files to Cell Ranger, and it does everything that is necessary for the upstream analysis of the data. So taking from the raw files to the alignments, then from the alignments to the count matrices, and the count matrices are where we're at this point. So I'll show you how Cell Ranger works. I'll show you where to get it. We're not going to be running it ourselves because I don't think any of us has a computer that could handle actually running a single cell experiment through Cell Ranger, but I'll show you how to do it. But if you happen to be Zooming from inside of a high-performance computer, yeah, or if you've got Threadripper, feel free to follow along in real time. All right. Yeah, if you were fortunate enough to manage to get a Threadripper CPU, it's pretty hard to get stuff like that right now. Okay, so let's go ahead through the analysis now. I'm assuming that all of you are looking at the same thing I'm looking at. If you're not, please do your best to pull this up while we're here, because I think it'll be really instructive for you to be able to run this as I'm going through it. Okay, so this first set of code here is basically asking a question. So hopefully you've all got a bit of basic familiarity with R, and we'll know the file.exists command. Basically, it's simply asking the question if a file exists or not. And if the file doesn't exist, it will return false. So let's take a look at what this actually returns. So it says false. This file does not exist yet. And so in our if-else here, or I should just say if here, we're saying not file exists. So we're basically asking the question, if this file does not exist, then do this. And if the file does exist, it's not going to do anything. But because it doesn't exist, it's going to download the file using download.file from this website. And then it's going to untar the file because the file is going to be a .tar.gz archive, which actually you can see right here. So you don't actually need to run this download.file command. You could actually just straight untar it. But this will work no matter how you have things configured, whether or not you actually have the data here. And in the future, depending on how large this the data for this workshop gets, I might end up removing this .tar.gz file from the GitHub repo. So if it's not there, this command will get it and untar it. So go ahead and run this. And you can just put your cursor anywhere in this line number 6, hit Control-Enter or Command-Enter, and it'll run. So you can see here it's downloading the file. Doesn't take long, and it untarred it. And then when it untarred it, it's now in this folder here. So .tar.gz is just a compressed archive, so not too dissimilar from a zip file. And when it expands, it turns into a folder. So you click on this, and you can now see inside, and you click on this, and you can see now the 3 items contained within: the matrix, the genes, and the barcodes files. Essentially what you're seeing here is the gene count matrix is a matrix.mtx file. And rather than actually having the row and the column names in the matrix, they put them separately in 2 different files. So these are the row names, they're the genes, and then the column names are the barcodes over here. When we load this data set using scRNA, it will automatically compile this data and put it correctly into one matrix. So what we're going to do is we're going to now load the scRNA library. Go ahead and do that if you haven't already. Just takes a second. All right. And now you can use the read 10x function, which comes from this library. And there's one argument that we typically care about, which is data.dir. And we're going to say the data is in this filter gene barcode matrices folder under the HG19 directory. So we're basically pointing it to this folder right here. So go ahead and run that. And it's going to read in the data and compile it into one matrix. You'll see that now in your environment, you've got this pbmc.data object. And so let's say we want to look at this. We want to see what kind of object is this? What does it contain? Go ahead and print it to the screen by just running that line 20. And you can expand this and scroll up to the top. And you'll see it'll tell you here, it is a sparse matrix of class DGC matrix. We'll get into it tomorrow, or not tomorrow, next week. More what a sparse matrix means and kind of why we use these and what it kind of implies about single cell data. But for right now, just know that this is not too different from a normal R matrix. It's just that when you have a 0, it's represented by a dot when you print it. We also know that's 32,000 rows by 2,700 columns. So that's 32,000 something genes by 2,700 cells. And there's a lot of zeros. Some ones every once in a while, but almost entirely zeros. Again, we'll get to that next week as to what that means, why it's like that, and what the implications of that are, because that is in fact a feature of single-cell RNA-seq data, and we call it zero inflation. So the next step is to actually create your scRNA object. You can easily do that with this create scRNA object function, giving the data matrix as the counts argument. And go ahead and run that. So that ran very quickly. It's given us a warning here. Feature names cannot have underscores replacing with dashes. So basically it said that the gene names that you gave it had underscores. So some of these genes had underscores in them and they just replaced it with a dash. You can pretty much safely ignore that. I've never really run into an issue with this. And then it is now created a scRNA object called PBMC. So if we print this, we can see it's a class scRNA object with, and this is basically just describing what we said before about the size of the matrix. And it says that that is being stored in the RNA assay. So this is something you might not know. If you have any object in R, you can easily examine it by clicking on it in the environment panel. So now I can see here it represents it as kind of a list. I can now see all of the elements that are contained inside the object. So the first element is assays. That's a list containing one element, which is called RNA. So down here it said the active assay was called RNA, and there it is. So if I come over here and click this, it allows me to directly access that RNA assay object. And I can print it to the screen and see some information about it. In general, you'll do all this with automated sort of functions that scRNA provides you, but it's just nice to know where the data lives and if you needed to, how to access and manipulate it. Because sometimes the function scRNA gives you will not be enough, and it'll be nice to know how to grab the data when needed. The next thing that I'll draw your attention to is this metadata object in here. And if you actually view that by clicking that scroll, you can see that it describes all of these cells. So there's 2,700 rows. So these are all cells here from your gene count matrix. and then it gives you information about them. So this is the metadata for your cells. If you know, for example, that you've got some patients that have Alzheimer's and some that don't, that would be one of your columns here, and you can easily add that data in after the fact. It's also telling you some really important quality metrics, which we'll talk about next week, called N-count RNA and N-feature RNA. So just bear those in mind. Next week, we will tell you how to use those and essentially what these mean. And that's pretty much it for this week. So I know we didn't get through a whole lot, but at this point, you should know how to get some data and how to load it up into R. So what I want you to do as an activity, and since we don't have a whole lot of time, you'll do this for homework, is I want you to find your own single-cell RNA-Seq dataset from 10x Genomics, and I want you to download it and load it into R. So if you go to this link here, I've included it in the script. You can find that 10x Genomics, which they are the makers of a very popular system for doing single-cell sequencing. You can go down to their gene expression datasets and start looking through and finding ones you're interested in. So there's lots of different categories they have here. I typically stick to the Chromium demonstration categories if I'm doing just an example data set, because they tend to be a little bit less complicated. So I'll stick to this one. The options you select here don't matter a whole terrible lot, but it does determine a little bit about essentially what genes might be there and how it might be formatted. But for right now, it's really not a huge deal which you pick. Just make sure not to pick one that's like 1 million cells, because it's unlikely that unless you've got a Threadripper, your computer will be able to handle that. So you can come down here. So these are 1,000 PBMCs from a healthy donor. So let's go ahead and take a look at this. And in fact, there's a lot of information here and a lot of stuff you can see if you want to. You can even get the BAM files or the FASTQ files. So this is the raw data. But typically what we want is this filtered feature matrix here. Now this is essentially the material that we're going to be starting with anytime we do a downstream analysis of single-cell RNA-Seq in R. And if you're looking for a data set to run as an example, this is probably a pretty good one to choose. So I would just download this. It gives me the tar.gz. I could also download it using the download.file function, and I could untar it using this. So go ahead and do that for practice. And I'm not going to check that you've done it, but it's a really good way to solidify the skills that we've talked about here. And with that, that's it for this week. So I'm just going to pause now and take any questions or any concerns. If you're not able to replicate what I'm doing here, feel free to unmute or put in the chat. And if you don't have any questions, if you feel totally fine with everything that I've shown here, feel free to drop off, and I'll see you guys next week. So someone asked, will the video be made publicly available? Yes, all of the recordings will be made publicly available. We're transitioning into a new system where we put them on YouTube, and we're actually making a website that Aola is spearheading that's going to contain the videos and the materials from the lessons. So once that's online, you'll get access, but you'll have access to the videos through Box. I'll make sure you all get the link. Okay, were there any other questions before we break? So Mauricio, I want to make sure I clarify what you said there. So are you asking for me to show an example of getting the data from a database? Okay, so I'm actually going to go through that in future sessions, because for right now, I just want to keep it simple. But in future sessions, probably I think in week 3, I'm going to show you how to do that. Because you're right, that is much more realistic to how you'll actually end up doing it in reality. Yeah. And that can be also a bit trickier. And yeah, we'll be using Zoom from now on. I'm going to send another email that just confirms that for everyone who wasn't able to make it today. But yeah, we'll use Zoom from now on, because we're not going to break the 100 participant limit, it looks like. Okay, well, I can't tell you all how much I appreciate you being here. Oh, okay. We got one more question. How to read data that are in different... Okay, so H5 is another really popular format for storing single-cell RNA-Seq data. And also, there's actually a built-in function in scRNA for reading from an H5 format that, since you've asked for it, I'll just show quickly, probably next time. And other formats, there's typically helper functions in scRNA to read from any format. But if need be, you can construct your own object from just like a TSV of like a count matrix. And I can also show you how to do that, because pretty much no matter what tool you were using before scRNA, you can at least get a TSV or a CSV out of it. So I'll show you how to do that if that's all you've got. Okay, well, I can't tell you all how much I appreciate this. It's really nice to be able to give back, and because a lot of people helped us when we were first learning. So, okay, I got one more question. Will we be learning potential subsequent conversion methods between different file formats? I'll go through a couple of them, and I'll tell you what scRNA functions are available to help you with that. I'm not going to do all of them. If you've got any that you want me specifically to go over, let me know, and I'll probably add those in, especially if we've got a good amount of time left over, because I agree that would be very useful. All right, so this concludes our first session. I can't tell you all how much I appreciate you being here, and I'm really looking forward to working with you all for the next few weeks. All right, you guys have a good week, and I will see you next time.